Particles are everywhere, especially in cities like Stockholm, where we have traffic on road, rail, air and sea. When the bus brakes, the engines run and the tires roll on the streets, small particles fly out into the air that we breathe. And particle emission causes millions of premature deaths every year. In the European project nPETS, we are targeting the smallest particles, sized as viruses that can go deep into our lungs. And these particles are not regulated by law, mainly because there is no standard way to measure them. So we don't know how many and how toxic these are.
We're in central part of Stockholm, and just next to us we have a road tunnel, and we're going to examine how toxic the particles from the air in the road tunnel are to us humans.
To assess the toxicity of the traffic emission, particles are sucked from the road tunnel and they are put into the model where cells on the bottom are exposed.
The nPETS project aims also at evaluating the average population exposure to nanoparticles in ambient air at 4 European cities. This is possible with this kind of mobile laboratory vans equipped with state-of-the-art instrumentation for the monitoring in real time of nanoparticle concentration and for the collection of samples of nanoparticles, which allows to perform physical, chemical and toxicological analysis in order to understand which sizes, which compounds and which sources are the most responsible of the toxicity of such nanoparticles.
Here we are collecting and measuring particles. We have this dilution system, and then here we have all the instruments that we can collect and analyze particles.
We are also involved in the chemical analysis of the nanoparticulates investigated within the project. In our laboratories, we characterize the inorganic fraction of specimens for laboratory tests of specific emission sources, as well as from environmental sampling campaigns.
But do the particles from the various sources differ in toxicity and health effects? In this lab in Stockholm, we remove the particles from the filters and then we expose cultured human lung cells. We want to find out if the particles can cause inflammatory effects and damage to the cell's DNA. Such effects are linked to several diseases and can thus be used to predict health effects.
Thanks to this unique collaboration between researchers from several universities and institutes from Europe, decision makers have knowledge so they can regulate harmful particles. Now it's up to them to decide how to use this new knowledge. Maybe we have to change our behavior and how we build our cities.